Hello guys and welcome back to Excelsior Academy. So today we are going to do the lecture 6 as part of the Watson Summary Series chapter the Mutability and the Repair of DNA. So in this lecture we are going to discuss about the repair and the tolerance of DNA damage. इससे पहले हमने DNA damage के बारे में पढ़ा and कुछ repair mechanisms भी पढ़े जैसे कि mismatch repair, right? So now we are going to study about the further DNA damages and how it has been repaired. So DNA damage can have two consequences. So some kinds of damage such as thymine dimer हो गया या nicks हो गया या breaks हो गए in the DNA backbone, they create impediments to replication और transcription. इसका मतलब क्या? अगर इस तरह के DNA डैमेज होते हैं तो वो आपका replication transcription अच्छे से नहीं होने देता, यानी कि एक तरह से it is not possible for the cells to undergo replication transcription if the DNA backbone is broken. ठीक है? Other kinds of damage create altered bases that have no immediate structural consequences on replication but cause mispairing. These can result in a permanent alteration to the DNA sequence after replication. तो हमने ये पिछले लेक्चर्स में डिस्कस किया था, लाइक फॉर एग्जांपल आपका AT है और जस्ट बिकॉज यहाँ पे आपका मिथाइल ग्रुप लॉस हो गया है सो इट बिकम C, सो देयर विल बी फ्रॉम AT to GC, राइट? तो इस ट्रांजिशन की वजह से व्हाट विल हैपन इफ इट इज not detected, so this transition will become permanent. Correct. अब जब ये permanent हो जाएगा, so the problem is that in the next round of replication the sequence will be different as from the parental sequence, and because of this there will be change in the gene sequence. अब जब आपका gene sequence change हो गया है तो most probably there are chances that it is going to get non-functional या फिर कुछ और ही म्यूटेंट प्रोटीन कोड करेगा. ठीक है? सो दीज आर द प्रॉब्लम्स इफ इट इज नॉट बीइंग रिपेयर्ड एंड इट बिकम्स परमानेंट इन द जीनोम. So this is what just now I have told about this. The conversion of cytosine to uracil by deamination creates UG mismatch, which after a round replication becomes a CG2TA transition mutation on one daughter chromosome. These considerations explains why cells have evolved elaborate mechanism to identify and repair DNA damage before it blocks replication or causes a mutation. ठीक है? Cells would not endure long without such mechanism. ये तो सही है भाई. अगर इतना सारा अगर DNA replication errors हो गया, बहुत सारी mutation हो गया, if these are undergoing without any, if these are not being repaired, तो क्या होगा? एक सेल के लिए हैंडल करना बहुत मुश्किल हो जाएगा इतने सारे म्यूटेशन. ठीक है? एंड इट विल नो लॉन्गर बी एबल टू सर्वाइव. इसलिए बहुत सारी रिपेयर मैकेनिज्म है. वंस इट इज नॉट एबल टू रिपेयर, एक बार रिपेयर ही नहीं हो रहा है, देन the cell will try, it will give itself a programmed, कि हाँ अभी नहीं हो रहा है तो it will undergo the apoptosis. ठीक है? Now in this section, we consider the systems that repair DNA damage in addition to the mismatch repair system that corrects mismatches during the replication. So let's talk about the types of the DNA damage and the tolerance system. So type हो गया, ये हम discuss कर चुके हैं, mismatch repair. So it is nothing but the error which happens during the replication, right? So what are the enzymes involved to repair? म्यूट एस, म्यूट एल, म्यूट एच इन इकोलाई एंड उसके होमोलॉग्स हो गए आपके MSH, MLH एंड PMS इन ह्यूमंस. ठीक है? फोटो रीएक्टिवेशन, दैट इज फॉर द pyrimidine dimers, यहाँ पे आपका thymine dimer बनता है. एंजाइम्स है DNA फोटोलाइज, so this is able to break the bond between the thymine dimers. बेस एक्सेशन रिपेयर, डैमेज बेस है DNA ग्लाइकोसिलेज, तो ये आपका पर्टिकुलरली दी ए बेसिक न्यूक्लियोटाइड जो रहेगा उसको रिमूव करेगा. कमिंग टू द न्यूक्लियोटाइड एक्सेशन रिपेयर, pyrimidine dimer हो गया या बल्कि एडेक्ट्रॉन बेस हो गया, so these will be removed with the help of the UBR A, B, C and D in E. coli. उसके होमोलॉग्स हो गए आपके XPC, XPA, XPD etc. in humans. Now double strand break repair, Rec A and Rec BCD in E. coli. Translation DNA synthesis के लिए हो गया आपके पास the pyrimidine dimer, A purinic side or the bulky addictome base by family of DNA polymerases such as HumuC in E. coli. Guys, ये टेबल बहुत बहुत इम्पोर्टेंट है. आप I really suggest कि आप भी अपने नोट्स में जो भी आप शॉर्ट नोट्स बनाते हैं to prepare for the CSIR exam, just उसमें ये टेबल बना ले and direct डिक्टो ऐसा का ऐसा ही लिख लीजिए because this is really very very important for you guys to remember. ठीक है? Okay. So in the most direct of these systems, a repair enzyme simply reverses the damage. There are two kinds of existing repair system which exist. One which involves the removal of only the damaged nucleotide, and the other which involves the removal of a short stretch of single stranded DNA that contains a lesion. ठीक है, तो यहाँ पे क्या हो गया, फॉर एग्जाम्पल अगर यहाँ पे आपका एक मिसमैच है, तो एक तरीका होगा कि दैट पर्टिकुलर न्यूक्लियोटाइड विल बी चेंज, ये एक दूसरा एक मेथड ये हो गया. दूसरा मेथड क्या होगा, दिस पर्टिकुलर सीक्वेंस विल बी चॉप्ड ऑफ. ठीक है, and then again using the template strand it will be resynthesized with the help of the DNA polymerase and DNA ligase, it will be sealed back. ठीक है, ये दो तरीके हो गए हैं. Yet more elaborate is recombinational repair, which is used when both strands are damaged as when the DNA is broken. So ये एक दूसरा तीसरा मेथड हो गया, the recombination method. यहाँ पे आपका अगर both strands of DNA are damaged, ठीक है, अगर दोनों double stranded break है या damage है, so best method to repair will be the recombinational repair. Of course, the DNA has to be homologous. In such situation, one strand cannot serve as a template for the repair of the other. Hence, in recombinational repair, which is also known as double strand break repair, sequence information is retrieved from a second undamaged copy of the chromosome. इसलिए मैंने बोला कि homologous chromosome की जरूरत पड़ेगी. See, in this case, we know that यहाँ पे हमारे पास homologous copies हैं, ठीक है, और दोनों break है. So what will happen? In order to repair this, we need another copy of the chromosome which is undamaged. हम उसके sequence information को use करेंगे so that we can repair in the first chromosome. So this is nothing but known as double strand break repair or recombinational repair. Finally, when damaged bases block progression of a replicating DNA polymerase, a special translation polymerase copies across the site of the damage in a manner that does not depend on the base pairing between the template and a newly synthesized DNA strands. अभी हमने क्या मेंशन किया, एक तरीका होता है जहाँ पे आपका जो डैमेज हुआ उसको रिमूव किया, डीएनए पॉलीमरेज आया, उसे रिपेयर किया, देन लाइगेज ने उसको ज्वाइन किया. ठीक है, अब दूसरा तरीका क्या रहता है, देयर आर स्पेशल डीएनए पॉलीमरेज व्हिच आर नोन एज द ट्रांसलेशन पॉलीमरेज. ठीक है, so what they do is that they come and they copy across the site of the damage in such a way that जहां पे आपको बेस पेयरिंग की जरूरत नहीं है. ठीक है, and this is happening as a mechanism of the DNA damage टॉलरेंस. पहले क्या हो रहा है कि आपका रिपेयर हो रहा है, सो रिपेयर के लिए यू नीड द टेम्पलेट सीक्वेंस इंफॉर्मेशन. अब क्या है, यू डोंट हैव द टेम्पलेट सीक्वेंस इंफॉर्मेशन बट यू कैन नॉट कीप योर डीएनए ऐसे इन द ब्रोकन स्टेट. सो इन ऑर्डर टू मेक श्योर दैट द सेल डज नॉट डाई, ठीक है, तो हम क्या करेंगे, इसको हम टॉलरेट करेंगे. सो दिस काइंड ऑफ डीएनए डैमेज विल बी टॉलरेटेड बाय द सेल सिस्टम. ठीक है, so it's a system of last resort because transition synthesis inevitably error prone, यानी ये mutagenic है. So it's like मरने से अच्छा है कि हम एक म्यूटेशन या एक डीएनए डैमेज हम टॉलरेट कर ले, so that अगर हमने अभी सर्वाइव कर लिया, so in the second round of replication that will be repaired. ठीक है, so this is the meaning of being tolerating the DNA damage. Now the first thing is direct reversal of the DNA damage. इसका क्या मतलब हो रहा है, that you have got the DNA damage and that will be because of the another mutation, it will go back to its original state. Okay, for example, photo reactivation. So this is required in the case of the thymine dimer. So it is an example of repair by simple reversal of the damage is the photo reactivation. It reverses the formation of pyrimidine dimers, जो आपका thymine dimer बना था, with the help of the UV light. Okay, with the help of the UV light, that is UV irradiation, this thymine bond will be broken. So in this photo reactivation, the enzyme DNA photolyase, it captures the energy from the light. It uses to break the covalent bonds linking the adjacent pyrimidines. Okay, so this is how the DNA photolyase is there, which comes and breaks the ring found between the two thymine dimer. तो यहाँ पे UV light रहा, आपका pyrimidine dimer है, DNA photolyase है, which goes in the dark, it takes the energy from the visible light and breaks the bond. ठीक है, then it is breaking and क्या होगा, आपका normal thymine आ जाएगा, which will base pair with the your adenine. So this is the direct reversal of the DNA damage. Now coming to the another example is the removal of the methyl group. It is removal of the methyl group from the methylated base O6-methyl guanine. In this case, methyl transferase, it removes the methyl group from the guanine residues. ये guanine residues हो गया, it has got the additional methyl group. Now this will be transferred to the cytosine. So this is costly to the cell because the methyl transferase is not catalytic. Having once accepted a methyl group, it cannot be used again. अब problem क्या हो रहा है? Look it over here. See, in the DNA damage, this case, you have got the O6 methyl guanine. यानी कि आपके ऑक्सीजन 6 ऑक्सीजन ग्रुप में methyl group आपका added है. So it is not possible to base pair with the cytosine. Now to remove this, methyl transferase is there, which comes and takes the methyl group to its own cysteine residues. ठीक है? अब क्या हो रहा है? The thing is, आपका DNA तो रिपेयर हो गया, and this is like covalently accepted. So the only problem is that this enzymes needs to be then degraded off. क्यों? Because it cannot be reused again. So अभी क्या करेंगे? This methyl group is there. This entire enzyme becomes the useless after it accepts the methyl group. So that will be degraded and broken down into its simpler components. Then these amino acids will be recycled. ठीक है? पहले कैसे होता है? अगर आपके पास ये ग्रुप है तो आप ये ग्रुप रिमूव कर देंगे and enzymes will be functional. But in this case, it is not possible. Okay, since it's not that catalytic. So once it has accepted the methyl group, it cannot be used again. So पूरा का पूरा enzymes हम degradation के लिए send कर देते हैं. Okay, so guys, that's all for today. So today we discussed about the two examples of to summarize, two examples of direct reversal of the DNA damage. पहला हो गया था आपका photo reactivation, and the second one is about the O6 methyl guanine. So guys, I hope you understood the topic and it was really very simple है, बहुत ही simple है, simple भाषा में समझाया गया है. I hope you remember the content because that is what is more important. So even if you hear for another 10 minutes, the point, the taken away message is that you should remember there are two different types of direct reversal of the DNA damage. पहला हो गया आपका photo reactivation where you're talking about the thymine dimer. एक दूसरा हो गया removal of the O6 methyl guanine and how to repair. तो आपके इसको enzymes के नाम भी याद रखने हैं. So in the first case it is the DNA photolase, in the second case it is the methyl transferase. So guys, इसी तरह से हर एक lecture के बाद just summarize what you have learned and make a short note of it. So कैसे होगा? The Watson chapter which you might have taken around 1 week to complete one chapter, by listening to these videos you can complete in an hour. Okay, so guys, thank you for watching till end and keep watching. Make sure you make your own notes also. So keep studying with us. Definitely we are going to excel together. Thank you guys. Take care.